We're going to move on to challenge our third challenge here, but then additionally add some uh, more color commentary on some other topics. But we'll get to our third challenge now, which is the lack of animal models and what does that really mean. So, many cell and gene therapies are intended for diseases and conditions that primarily impact pediatric patients, like we talked about, requiring the prospect of direct benefit to individuals and subjects that are enrolled in the trials. But in the absence of good animal models, there may be challenges demonstrating the prospect of direct benefit. So, Nina, let's just stick with you if that's okay. What are these novel approaches to consider? And what are the challenges due to often the lack of animal models to help really effectively validate new technologies?
Yeah, so I think we definitely need to look at novel technologies for an answer here, especially as we get down to specific mutations and, like, just call them gene editing, for example. Like, how will we be able to demonstrate a lot of that prospect of benefit early on for some of these approaches? So, for example, organ on a chip, an organ chip, microfluidic culture devices are perhaps promising in vitro systems that can be used to recapitulate organ level and even organism level functions. And so more use of that and for the agency to learn more about their use, I think would be really helpful. Some those devices, they can recreate organ level structures that will hopefully model organ physiology and disease states for some of that demonstration. So body on a chip, for example. We also need to work together to identify the needed information to validate the use of these for clinical development. And so this is where sometimes, for example, FDA's innovative science and technology approach for new drugs is a pilot program that was really designed to encourage the development of drug development tools that are out of scope for qualification programs, the longer term biomarker clinical outcome assessment programs, but to more focus on other areas that are still beneficial for drug development, for example, organ chips. And even when there are animal models available, there still are challenges, right, in terms of how that really represents what might happen when you go into a clinical study. And there was a meeting, I think maybe in the end of 2019 now, where FDA focused specifically around sort of fit for purpose, what animal models to use and to think thoughtfully about why you're using them and sort of what answers you're looking for, because even existing models won't be able to provide all of that information earlier on.